Thanks, Debayan and Sandeep, for organizing this and inviting me in this workshop. So, my research is on plasma membrane phase separation, which is a little different than what we are typically thinking about currently about LLPS using proteins and nucleic acids, which has a lot of interesting chemical properties, such as they might be involved in cation pi interactions and different functionalities, pi interactions. But lipids, on the other hand, are not necessarily have all these diversities. Lipids are, like as we all know, it's an amphiphilic molecule. It's a head group, and then it has a long fat. But what I can show here today is that the phase separation of these lipids in the plasma membrane plays big role in the functions of cellular signaling, and in this time, the signaling that I'm interested in, immune receptor signaling, that happens, and all these measurements are done in living cells. So, the immune signaling that I am interested in is allergy signaling that happens in our body. So, in allergy signaling, what happens once we are exposed to an allergen, say for example, brinjal or peanut butter or whatever, and then this particular receptor called Fc epsilon R1, this allergen binds to them, it gets clustered, and then a lot of processes downstream to this starts working. In the end, it releases some materials called histamine, and we get those small little blisters, red blisters, which are moieties packed of histamines, and then to inform you that you have been attacked by an allergen. And then you take an antihistamine medicine to reduce it, and you are good to go. So, just focusing on the very early stage of this, what happens is that the Fc epsilon R1 receptor binds to the antigen, it makes a complex, then this complex is not active. Active means that once this complex is phosphorylated, remember the antigen is bound to the outside of your cell and the phosphorylations happen at the inside of your cell. So, which means that antigen cannot directly phosphorylate the receptor. So, something must have happened inside to phosphorylate the receptor so that all these downstream processes can start. And this happens by in all biological processes, phosphorylation is catalyzed by a molecule called kinase. And of course, in biology, everything must be reversed because you take a medicine, it must be reversed, right? It has to be reversible, and there is always an antidote to kinase, which is a phosphatase. So they are all present there. Now the question is that we are putting here is that how this receptor, kinase, phosphatase, all of they are present before and after antigen, but this activity is happening only after antigen is present, right? Because everything is happening in the intercellular part and they are all present in your body at all times. But the moment something is bound outside, it is doing something and we start seeing this activity, or in other words, this balance is shifted more towards right so that the signaling happens. And this is where the an attractive model that comes in is an LLPS model. So, and it is the LLPS of the lipid. So, what this model posits, it states that when you have unbound states, we have nothing, and then when the receptor is cross-linked with the antigen, it forms this cluster, and this cluster somehow assembles an ordered state around it. Now, when you said ordered state, I'll be discussing a little more in the next slide. In ordered state, what happens is that there are lipids which has unsaturation in their acyl chain in their tail, so they do not pack well, and then there are lipids which are saturated lipids and they do pack well. So, you see that if these lipids are put together, there is no packing defect, and if these two lipids they self assemble, there are packing defects. So, this makes them more disordered, and this makes them ordered. Now you mix them. Mix a lipid which has unsaturated tail, a lipid saturated tail, along with cholesterol which is ubiquitous in plasma membranes. And then you start seeing a unique behavior where cholesterol does break this long range attractive interactions in these saturated lipids, and also it induces some sort of order in this unsaturated species. So giving rise to a liquid phase separation at the plasma membrane. So this is what you see in the model system. So once we get that, now the kinase, which is lin over here, this is a protein that is anchored to the plasma membrane in inner leaflet by a saturated tail. So of course the kinase likes to be present, so it partitions into those liquid ordered region. So now it has a more chance to see the receptor and the receptor tyrosine, and it can do its action. Okay, so that is the model. So moving to the membrane lipids, membrane organization, you see that there are inner and the outer lipids. This is the outer leaflet and the inner leaflet. There's a very diverse lipid composition, and if you, as I was talking about the tail unsaturation, you see that outer leaflet has a lot of saturated species, while the inner leaflet. So when this x-axis is lipid unsaturation, meaning that most of the lipids in the outer leaflet are indeed saturated. There is zero unsaturation compared to the inner leaflet, where most of the lipids are unsaturated. So, so that's one thing. The other thing is that is also experimentally tested, and it was found that outer leaflet, since it has a lot of less unsaturation or almost mostly saturated, it has a slower diffusion. As I said, it is more ordered, hence it has slower diffusion compared to the inner leaflet, which has a faster diffusion. Similarly, you can look at the other. So this is the direct measurement of lipid order, and you can confirm that. So what happens, as I was mentioning, that there are unsaturations over here, saturation, and then when you put them together, you can really create this LOLD like membrane. This has been tested experimentally by standard phase diagram. It's been long tested, DOPC, DPPC, and cholesterol, and all these are not in live cells. These are like synthetic lipid membranes. So you can clearly see what we call nowadays a familiar word, droplet, right? So you see over here, this is a fluorescence image, and of course outside this coexisting region you have a single phase, and as you go from a little bit inside this boundary or above the CSAT of all these three components, you start seeing phases. So go from 4, 5 to 7, you are increasing the cholesterol content. 4, 5 to 7, 4, 5, 7, you are seeing more and more phase separation. 4, 6, 8, you are getting more saturated lipid content. 4, 6, 8, you are also getting more phase separation. All right. So this is all good, and again on them people measure diffusion directly, and they say differences. So yes, both of them, the tracer molecule, they do diffuse in both phases, hence they are both liquid, but they diffuse at a very different rates. So one is in the order of 0.1 micrometer squared per second, one is about 3-4 micrometers squared, so order of magnitude difference, and which means that both of them are liquid but have a very different diffusivity of the tracer inside those domains. So these are all good in synthetic membrane. The problem starts when you start looking into it in live cells. So these are live cell images. So this is your cell, and in the cell you do not see any phase separation. But what are those? So we create chemical stress into the cells, and the cells set off some vesicles directly from the plasma membrane. Now the moment the cells are shedding this plasma membrane, the vesicles from the plasma membrane, you see that there are phase separation. So in other words, the hypothesis is that maybe the plasma membrane do phase separate, but they are not resolved by our optical microscopy. But when you are trying to get the vesicles directly from the plasma membrane, or in other words, the cell is totally in out of equilibrium. There are so many things happening in cells. They are full of ATPs, there are actin cytoskeletons, there are other things that are controlling the behavior of the plasma membrane. But now you are isolating a vesicle directly from the plasma membrane, which does not have all these complexities, and you see now you clearly see the vesicle. So in other words, it is allowing these small little droplets to coalesce so that they become optically resolvable. So all in all, this image states that plasma membrane is likely have the capacity to phase separate. And indeed, when we took these vesicles directly out, and here what we are doing in the cytoplasmic leaflet, we take different probes, saturated lipid probes, unsaturated lipid probes, and you see that they clearly phase separate. Okay, so just now, which means that the inner leaflet of the plasma membrane, when this is isolated from a living system, they do phase separate. Again, the diffusion behavior shows that they're liquid, but they're in a very different state. So now the question, so this gives us hope that we can test whether this phase separation impacts our interactions between the kinase and the receptor, as I was hypothesizing in the couple of slides back. So what do you see now? First, we start testing by standard imaging whether or not we see the nanoclusters. Yes, we do. So you just stimulate the cells. Before that, there is almost featureless membrane. This is the receptor that we are imaging. And after you add the antigen, you see the small little dots, which means the receptors got clustered. Then, if the signaling happens, meaning that the action between lin and kinase happens well, then the Syk kinase, which was at the cytosol, that will go and bound to this receptor, and you will start seeing them at the plasma membrane in a similar punctate features, which is what you see, which means that the signal has been transmitted from outside to inside, and Syk got to know this, and it is recruited to the plasma membrane, which also means that there must be an intermediate step in which the receptor got activated by the kinase. So if there is a clear interaction between the kinase and the receptor, we are supposed to see similar punctated structure of lin kinase as well. And why is that so? Because if they are complexing, if they are talking to each other, if they are forming colocalizing at all times, then I am supposed to see the punctate structure, just like I see for the Syk, but I don't. It means that, but given that I am already seeing Syk is recruited to the plasma membrane, it means that this interaction had happened already. But this is a featureless image. It means that this interaction must be very transient. And this is also observed when we do the standard FAB experiment, in which what you do, you just bleach a spot into the plasma membrane and you see how the molecules are recovering. So if a molecule or a protein is immobile, it doesn't recover because it doesn't diffuse anymore. So that's the case for the receptor, because it was bound, it is clustered, and it is mostly immobilized. Lin, it's not the case. It was mobile before receptor clustering and after receptor clustering, meaning that it interacts with the receptor, but it is not strong enough to make it immobile. And this is how we see it. So let's say this is the lin molecules, the black ones, and it's the lin trajectory before and after clustering. So before clustering, there was nothing; it is not interacting with the receptor. But now the receptors clusters are formed, the larger ones, and the lin molecule are going there, interacting with the cluster for a while, and then coming out. Right? There are some lin molecules which will not interact with the cluster either. So now if someone makes a measurement in these particular regions, then they probably would see a little bit decrease in the diffusion of lin because it is interacting for a short while. But if someone is making a measurement on the other places like these ones, you don't see any changes of lin diffusion because they are not interacting. So in FRAP, I don't get to see it where I should bleach my spot. I don't know. So I just bleach anywhere randomly, and then I collect the statistics and I see, oh, nothing is happening, simply because we're not collecting enough. Maybe if we collect tens of thousands of data points, statistically you will probably be able to see that. And this is exactly what we need to do and we came up with this method that we do. It's called FCS, fluorescence correlation spectroscopy. This is another equivalent measurement of FRAP, but now you can get correlation function or diffusion measurement at each spot on the plasma membrane. These spots are very small, like 200 nanometer spot, and your membrane is 50 micron by 50 micron. So in one go, you get 10,000 data points. Once you do that, what happens? That indeed, you see that this is the cumulative distribution of diffusion constant of Lin, and the red one is before antigen. After stimulation, you see a clear red shift, blue shift in its diffusion constant. It means that the diffusion indeed decreased. Now, this decrease is only about 10%, which is what you expect, because remember I was telling the interactions is very transient. So, if it is a transient interactions and you are making a bulk measurement, you are not getting to expecting a huge difference, right? And this is and the good thing about doing this enormous measurement, so we are talking about 50,000 data points. When you do this scale of measurements, you start picking up small differences. Of course, you are now have the other problem that you end up getting false positives. So you do run a lot of statistical tests, which I'm not going to talk, but a lot of different statistical tests to and complementary experiments to test whether this is real or not. So that is different. That is, all these controls had been done to justify that this is a real 10% difference in the diffusion constant. Now, this is good. We got to see for the Lin. Now, the next question is, my original hypothesis was somehow after clustering there is a stabilization of ordered and disordered region. Around the cluster there is ordered region. So how do we test that? So we again go back to our standard probes, which is a saturated probe and unsaturated probe, and let's say this is their diffusion constant to start with. Of course, the saturated probe will have a little bit slower diffusion than the unsaturated probe. Now, if the stimulation leads to a stabilization of the ordered disorder, what will happen? This probe, the saturated probe, will spend more time into this ordered domain because the ordered domains are now stable, slightly more stable. So it spends more time in the ordered region, so diffusion further decreases. Now that is what I was telling, or what the first speaker of today described as above CSAT. So there is dense region in one place in exchange of a dilute region elsewhere. So if my ordered lipids, the saturated lipids, comes closer to the receptor, that becomes ordered and saturated rich region. Elsewhere, it becomes unsaturated rich region or saturated diluted region. So there, where the unsaturated lipid prefers to partition, it goes there and spends more time there, its diffusion increases. So if my measurement shows that PM goes, which is the saturated lipid probe, PM goes down, its diffusion, GG goes up, I know that there is an the LLPS has been stabilized. This is exactly what we observed. Again, these are the two probes I am showing now. I have a series of probes, saturated, unsaturated, where we saw the same thing. Next, what we did, okay, so we proved that we developed a method that detects very subtle differences. That method also detects that there is a stabilization of order and disordered phase separation in the plasma membrane. Now the next question, does Lin kinase utilizes it? Because remember, Lin kinase has all the protein motifs that directly interacts with the receptor, that can, right? So what we did for that, we created a mutant in which you see this lipid part, lipid component of the whole protein is mutated, and now we added another component that is disorder preferring. So while all the other motifs remaining the same, meaning that this Lin kinase remains functional, okay, only thing we changed, it is membrane attachment. We make it order preferring Lin to a disorder preferring Lin. And there are tests to show that that this become disorder preferring. It is just you add mild detergent to the system. If it is entirely soluble, we call it disorder preferring. If it is insoluble, we call it order preferring. Now, what we see here, we lost this stimulated decrease of lane. In fact, it exactly resembles what disorder preferring probe would do. It slightly increases, not as much as the other probe I showed, but it slightly increases the diffusion. So it is spending more time in the disorder region, even though it has all the protein interacting motif. So this is the example to show that our LLPS is implicating biological interactions. Moving even more detailed functional interactions, what we did here, we reconstituted in a system the kinase, phosphatase and the receptor, and we look at plus or minus Ag and see whether the receptor is phosphorylating or not. So, when wild type kinase, wild type phosphatase and the receptor all are present, we see clear phosphorylation after antigen. When we take S15, which is the lean mutant, we do not see anything. We do not see the change in phosphorylation, and of course, without lean, no phosphorylation. This is a control, negative control. Without lean, just with the phosphatase, no phosphorylation happen. So this is to show that just by changing the lipid anchor, not the protein interacting motif, we can actually change its function, and that's why I just mentioned that liquid-liquid phase separation is impactful in the functional of membrane. Okay, so this brings me to this another state of this another part of this project where we are looking at so initially in the resting states everything was present, and in the stimulated state LOLD is stabilized. So if it so happens, if I change anything in the resting state, I increase or decrease the order of the resting state, does it impact on the stimulated changes? Now, I would say yes, because in the end of the day, it is how much stabilization you are making over here that dictates how much time lean will spend over here. If the ordered region are not so stable, lean will not spend so long in there, and it will have less chance to talk to the receptor, and the vice versa. So I would say that if I make my system more ordered to start with before antigen stimulation, I will see a difference, or less ordered, I will see an opposite trend. So so this is what we did in this setup experiment. What we did here, we come up with an idea of exchanging the outer leaflet by by some lipids of our choice, and this lipid can be saturated lipid like DPPC or DSPC, or can be an unsaturated lipid. So once we do that exclusively on the outer leaflet, we see changes happening in the inner leaflet, and this is how the changes are made. You see that when again the probes are same, PM and GG. When we add the POPC, a unsaturated lipid, we see a diffusion to increase, and when you have the saturated lipid, the diffusion decreases. It means that if you add unsaturated lipid or disorder promoting lipid, you see disorder in the inner leaflet. Remember, all my reactions are happening in the inner leaflet. So I cannot perturb the inner leaflet lipid. So that I have to keep intact. All I am doing something in the outer leaflet so that I can induce order or disorder in the inner leaflet and test whether it matters. So this is what we do for PM, and similarly when we look at GG, we do the same thing. So there is a global change, unlike the last one when we see that somewhere it is ordering, somewhere it is more disordering. This time, since I am changing the outer leaflet in a global scale, I am seeing a global impact. Either the membrane is entirely becoming ordered or the whole inner leaflet is becoming disordered. And once so my system is now prepared before stimulation in a resting condition. The system is prepared either disordered or ordered. And now I add my antigen and see what happens to this sick recruitment. So sick recruitment is our functional test. So higher or more sick recruitment or faster sick recruitment testifies that it is over signaling, or if it is less sick recruitment or slower sick recruitment, we call it hyposignaling. Okay, so now we see in here that you see that, so initially we have the control that is in red. If you have POPC, which is disorder preferring probe lipid, you see that there is a decrease in the stimulated sick recruitment, meaning that again, how my system is prepared, how my resting state is prepared, that dictates whether my stimulation will be optimal stimulation or hyper stimulation. So with this, we end to this talk, and we showed that the plasma membrane lipid phase separation plays a big role in the biological context, and in principle, there is a functional impact of this LLPS, and again it signifies the importance of the resting state. If your resting state is poised to respond, then you will get optimal signal. If it is not, if it is somehow perturbed, then you also get dysfunctions. Okay. With that, I'd like to thank everyone who contributed, especially my collaborators and my lab members, and thank you for your attention.
I had two questions.
Yeah.
The first is, how do you deal with enzymes that flip lipids across the two leaflets?
Yeah. So the lipid flipping that we like, the time scale of these measurements are very short. So we do antigen stimulation, and within 10 to 15 minutes, this is all done. Lipid flipping, like the real, like the PS, phosphatidylserine or PC lipids, that flipping happens in the hours time scale. So we have tested that whether during this process of our measurement time scale, whether the lipid flipping is happening or not. Then we observed that lipid flippings are not happening, and those enzymes, they remain active. So they are doing their job, but it is not flipping.
Slow time scale.
Yeah.
The second question, which is, I wonder if you have heard of T cell independent B cell activation of naive B cells, where the major histocompatibility complex is not involved.
Yeah.
For instance, non-proteinaceous antigens like lipopolysaccharide.
Yes.
Where the epitope is actually repeatedly displayed on a very long chain antigen.
Yes.
That ends up cross-linking the immunoglobulins receptors on the cell surface.
Yeah.
From when I first heard of this, of course, liquid phase separation was not being talked about, but just listening to your talk made me wonder whether T cell independent B cell activation actually involves a clustering of receptors and any kind of LLPS.
Yes, the answer is yes, because and I know a couple of people who are working on that, exactly on that. The reason why I'm saying this yes is because all these components like lean, seq and other things, and the receptor and the multivalency, these are pretty much similar between B cell receptors and the mast cell receptors, the epsilon receptor. The differences is on how you present the antigen. Now, in the T cell independent B cell signaling, the presentation of antigen is very similar to that of mast cell. So that's why I think, and when I talk to those people who are working on that, they also see similar behavior, the LLPS of the lipids playing a role in there.
Very nice talk. So I had 3 questions. So one being, so how fast are the dynamics of the formation of these? You said within 10 minutes you see.
Yes. So that is exactly what we are now testing. It happens in the time scale. So after you stimulate, it happens in the time scale of 5 to 8 minutes. So that's what
And if you keep it for longer, do they coalesce to become like bigger?
That is what we don't see, and this is why this goes back to that small image I showed, that there are ways to stop from over coalescence, and these ways, one of the major ways is actin cytoskeleton, because these are meshwork and it doesn't allow you to grow. So if you deplete actin cytoskeleton, it is not optically resolvable, but if you do super resolution microscopy, and I have some data on that, you could see that they are phase separating in when the activity of actin cytoskeleton is diminished.
And if you increase the orderness in the leaflets, do the size of these things increase? Have you heard that?
I would think yes, but I never tested
Still
that.
diffraction limited.
It is still diffraction limited. I don't see it definitely. And one thing you have to also note that just by changing this order disorder, you are not changing the order on the resting cell, you are not stimulating anything. So you see no recruitment. So they are not necessarily happening at a larger length scale that the seek is fooled to be recruited. So that doesn't happen.
But even at smaller length scale,
I do believe that it is there some they are becoming larger. I do believe, but I haven't resolved. I don't know how to resolve it. Yeah.
PDF that you show.
Yeah.
Saying that they're different.
Yeah.
It's not very different.
It is not very different. But what we did is there are many various ways we tested. So what we did, we take these measurements and we just then take subset of 10% of the data, right, in both conditions. Only 10% of the data. And then we do all sorts of rank correlation analysis to say whether or not they are difference, only with 10% of it, and we do repeat it like 100,000 times and see how many times it is giving us totally different. And we say they are different if and only if after doing 1,000 times, every time it states the p-value is different using rank correlation.
Yeah. Well, I mean, generally, I have suspicion about those statistical methods.
Yeah. So
There are issues with
there are issues in, there are fundamental assumptions to it.
say p-value, you are assuming that the distribution
Is Gaussian. No, I am not doing. So that's why I said I did rank correlation, and it is.
but you believe that it is different. I mean,
And because I have experimental tests, plus all this, as I said, it's only the 10% of all data I took from both population and see whether they are different or not. And then this is done 1,000 times. And every time if I see that there is always difference, it is giving, that's the benchmark. But again, like some days later, there will be some better statistical test will come, and I will run through my those tests with my data. Yeah.
do you actually see like linear, like when you plot square average over time, you see linear.
To the range I am talking about, the length scale, yes, it is linear. Yeah.
Like
The length scale, remember, I am not looking at single molecules. I am looking at a length scale which is about 200 to 50 nanometer big. So at that length scale, the anomalous diffusion oftentimes are not observed. So I am taking all the possible anomalies together and they are averaged out at that length scale.